I welcome you back to another important lecture. And if I were to recall some of the things from the previous classes, so we have looked on to the ultrastructure of an animal cell and the plant cell and the bacterial cell. So we must not forget about these acellular particles that we have in our environment, and these are the viruses with us. Now you see, whenever we have to regard something as living, so it is important that they need to have this living mass in their cells, and that living mass is what we regard this, or we call this as a protoplasm, we say. And whenever we say the word protoplasm, so whatever approach is that there has to be the presence of nucleus. After nucleus, there must be the presence of this cytoplasm, and along with cytoplasm there has to be the presence of cell surface membrane. And if these three components would be available within a cell, so it has a living matter, so it would be living. But unlike the bacterial and the plant cells and the animal cells, so these viruses, they do not have any of these living components. Like they don't have any nucleus, neither they have any cytoplasm or the cell membrane in them. So that is the reason we regard them as the non-cellular particles, and they are not living organisms. If we were to be talking about at what size these viruses they have, so even they are smaller than the prokaryotic cell. Now, what we know in terms of the size is that the eukaryotic size, cell size is the max among the living organisms. Then comes the bacterial cells, and at the end we have these viruses, and they have a diameter that range in between 20 to 300 nanometer. And if we were to be talking about the structure which they have, so in the viruses, we would have at the center would be their core, and in the core there would be the availability of the genetic material. And that genetic material in the viruses could be the DNA or the RNA, and this DNA and the RNA they both would not lie at the same time. The viruses that have the DNA in their core, so they are DNA viruses, and those viruses that would have the RNA in their core, so they would be RNA viruses. And all of the viruses, they have got this coat outside of them, and that coat is referred to as the capsid, we say. And every small unit that would be making this capsid of the virus, so we refer to this as the capsomere, we say. And rarely this so happens as like some of the viruses, they are regarded as the enveloped viruses because outside of their protein coat, which is made up of the capsid, so they would have the envelope around them, and that envelope again would be made up of the phospholipid. The reason is that whenever these viruses they would be attacking on some of the host cells. So when they escape through that host cell, so ultimately they would wrap themselves up with the membrane of that host cell, and such viruses they are the enveloped viruses. And not all the viruses are enveloped, but some of the viruses they would be. And likewise, some of the viruses they also have got these protein molecules in them, and these protein molecules they basically allow these viruses to attach with the host cell. Now the idea is that if the viruses they just have this nucleic acid core and they just have the protein which we were referring to this as a capsid, then how come the viruses they would survive, or how come they could increase their number? So the idea is very simple, that the time viruses would attack its host cell, so what it basically do is that it would hijack the protein building machinery of the cell. And by hijacking the protein building machinery of the cell, what we mean is that these viruses, after they have injected their genetic material into the host cytoplasm, so they would incorporate their genetic material with the host DNA, and by that they would hijack the ribosomes and the protein machinery or the protein making metabolism of the cell, and they would compel that host cell to be making the viral capsid and the viral DNA and the RNAs, and later they would assemble that together within the host cell, and ultimately afterward, this would escape out of the host cell. So this is what you need to know for your AS biology in context of the structure of the viruses. I hope you would be clear with this.